overview of the presentation. First, we will see the introduction, then what is DNA, then the operations that are performed in DNA, and how DNA can be used as a data structure. Then we will see the difference between DNA and the silicon-based computing. Then we will see Adleman experiment. Adleman was the person who gave the idea of DNA computing. And after that, we will see the disadvantages of DNA computing. Okay, introduction. As I have already stated, it was developed by Leonard Adleman at the University of Southern California in 1994. It illustrates the possibilities of using DNA to solve a class of problems that is difficult or impossible to solve using traditional computing methods. Uh, when we talk about DNA computing, the most important feature of DNA computing will be its massive memory capability, massive memory, and the capability to perform parallel operations, massive parallel operations. These two uh, would be the best advantages of DNA computing. Okay, let us see what is DNA. DNA stands for deoxyribonucleic acid, and it is the primary genetic material of all living organisms. When we say it a genetic material, we mean that everything related to a organism is contained in its DNA. So, for example, what is the color of our skin? What is the color of our eyes? In fact, what would be our height? Everything is coded inside the DNA of our body, inside the DNA of our cells. Okay. From here, the picture here, we can see that there is a cell. Okay, we know that human cell is very microscopic, and we need a electronic microscope to see it. Within the cell, there is a cell organelle called the nucleus, which is at the center of the cell, and within the nucleus there are chromosomes. Human beings have 32 pairs of chromosomes, and within the chromosomes there are DNA. So we can understand what is the size of DNA and how microscopic it is. Okay, a molecule composed of two complementary strands that are wounded around each other in a double helix formation. DNA has a structure of a double helix formation, as we have seen. In a DNA, there are basically four bases: A, T, G, and C. A stands for adenine, T for thymine, G for guanine, and C for cytosine. So basically, it is like adenine gets connected to thymine and guanine gets connected to cytosine. A connects to T, T connects to A. Similarly, G connects to C and C connects to G. We will not see any case where we will see adenine connected to cytosine or guanine, or thymine connected to guanine or cytosine. So it is always like A connected to T or T connected to A, and G connected to C and C connected to G. The complement for A would be T and complement for G would be C. Similarly, complement for T would be A and for C would be G. This is the structure of DNA. As I have already stated, there are four bases: A, T, G, and C, and we can see that A is only connected to T and vice versa, and G is connected to C and vice versa. Okay, now coming on to DNA operations. What are the operations that are performed in a DNA? The first one is annealing. Double-stranded DNA strands are dissolved in a single strands. This is called denaturing. Heating breaks the hydrogen bond between complementary strands, and this process is annealing. The next one is hybridization. Hybridization is base pairing between two complementary single-strand molecules to form a double-stranded DNA molecule. The next one is ligation. Joining of DNA molecules together is simply ligation. The next one is replication. As the name suggests, we can infer the meaning of replication. Replication means to make duplicate or to copy itself. So DNA generally replicates itself, and since the DNA replicates, the cell grows, the cells replicate as well. And this is how the human body grows. The next one is shorting. Shorting is arranging of things in a particular way. So in DNA as well, we sort the DNA molecules based accordingly on their size. Okay, the DNA molecules are basically negatively charged. So when we pass DNA molecules through say voltage, then we will observe that the smaller DNA molecules will get attracted towards the positive voltage, and the larger DNA molecules will get attracted towards the negative voltage. Now the next one is filtering. Filtering is the process of filtering DNA molecules based on their size. Again, various chemicals are used to filter the DNA. So these are the operations that are performed in DNA. Okay, how can DNA be a data structure? We know the massive memory capacity of DNA, and also uh, in traditional systems, we see that we have only two combinations, 0, 1, and we do all our computations based on these two numbers. But when it comes to DNA, we have four bases, A, T, G, and C. So this increases our ability for computation. Okay, in DNA replication, there is one error for 10 to the power 9 copied bases, or in other words, an error rate of 10 to the power 9, minus 9, sorry. In comparison, hard drives have read error rates of only 10 to the power minus 13 for Reed-Solomon correction. Okay, let us now see the difference between DNA and silicon-based computing. When it comes to storage media, we know that DNA computers have massively huge capacity of storage, and the media here would be nucleic acids. For conventional computers, it would be semiconductors. Memory capacity. DNA computers will have ultra high memory capacity, and conventional computers do have high capacity, but when compared to DNA computers, they are less. Type of operations. When we talk about DNA computers, there are biochemical operations, and we have seen the operations that are performed in DNA. These are annealing, hybridization, ligation, replication, sorting, and filtering. And when it comes to conventional computers, there are logical operations, and or not, or like these operations. And these operations can be performed on only two inputs, that are 0 and 1. And when it comes to biochemical operations, we have four base, A, T, G, and C. Nature of operations. When it comes to DNA computing, we have a massive capacity of parallel operations. And in conventional computers, everything happens sequentially. Although we have now some systems which do allow some sort of parallelism, but when compared to DNA computers, the parallel computation in conventional computers is few. Speed of operations. Okay, for DNA computers, it is slow, and for conventional computers it is fast. But the advantage of DNA computers will be that there are certain problems which till now conventional computers cannot solve, but these problems can be solved by DNA computers. So although it has a slow speed of operation, but it it can solve some problems which cannot be solved by the conventional computers. Adleman experiment. Leonard Adleman is the inventor of DNA computing. As I have already stated in the introduction slide, Adleman demonstrated a proof of concept use of DNA as a form of computation which solved the 7-point Hamiltonian path problem. So, in the actual research paper of Adleman, he had shown 7-point Hamiltonian path problem, but here we will consider only 5 point for the sake of simplicity. Okay, let us see the problem statement. Suppose that I live in Los Angeles and need to visit four cities, Houston, Chicago, Miami, and New York, with New York being my final destinations. The airline I am taking has a specific set of connecting flights that restrict which route I can take. That is, there is a flight between Los Angeles to Chicago, but no flight from Miami to Chicago. What should be what should my itinerary be if I want to visit each city? Okay, this is a Hamiltonian path problem. There are five cities, and Los Angeles will be our starting point. New York will be our stop point, and we have to travel through all the cities exactly once. So, what would be the procedures to solve the problem according to DNA computing? The first one would be to generate all possible routes. The second would be select itineraries that start with proper city and end with final city. The third would be select itineraries which with the correct number of cities. Here the correct number of cities would be 5 because we have taken 5 points. The fourth would be select itineraries that contain each city only once. So basically this is the Hamiltonian path problem. And now we will see each step how we can solve this problem using DNA computing. Part 1. Encode city names in short DNA sequences. So for each city, we will have a combination of DNA bases. Combination of DNA bases. So we have four bases A, T, G, and C, and we can use these four bases to represent each city. Here we have used six bases to represent each city. We can see here in the table, and how we are going to encode the routes. Encode routes between cities by encoding the complement of the second half last three digits of the departure city and first half first three letters of the arrival city. So for example, if our departure city is Miami and our arrival city is New York, so the DNA code which we have written using the base pairs for Miami is CTACGG and for New York is ATGCCG. Now it says that for the departure city, last three digits we will complement it. So for CGG, the complement would be GCC, and for the arrival city, we will complement the first three letters. So for New York, the first three letters are ATG, and their complement would be TA and C. So in here, this Miami to New York. So the coding for route encoding for Miami to New York would be GCCTAC, and likewise we will code encode all the possible combination of cities, all the possible combination of path and the corresponding cities which will come in between the path. The second step would be to select only those routes which has Los Angeles as the start point and New York at the end point, because Los Angeles is our starting point and New York is our final destination. So the second step would be to select only those combinations from all the possible combinations, to select only those combinations which have Los Angeles as a starting point and New York as ending point. And this can be done using polymerase chain reactions. These are the DNA operations. The third part would be to select itineraries that contain the correct number of cities. Since it is a Hamiltonian path problem, so each point needs to be visited. And since there are five cities, so the correct number of cities would be five. Now, in the second step, we are filtering only those combinations that has Los Angeles at the starting point and New York at the ending point. And in the third step, what we will do is that we will only select those combinations which has five cities. The fourth point would be to we have select itineraries that have a complete set of cities. So it says that we will select only those combinations that have five cities, and when we say that it has five cities, it should have each city once. We will select only those combinations, and this can be done using affinity purification. Successively filter the DNA molecules by city, one city at a time. Since the DNA we start with contains five cities, we will be left with strands that encode each city once. DNA containing a specific sequence can be purified from a sample of mixed DNA by a technique called affinity purification. This is accomplished by attaching the complement of the sequence in question to a substrate like a magnetic bead. The beads are then mixed with the DNA. DNA, which contains the sequence you are after the hybridizes with the complement sequence on the beads. These beads can then be retrieved and the DNA can be isolated. We can see this in the figure below, how it can be separated. Okay, this is gel electrophoresis. From this figure, we can see that, as I have already stated, that DNA molecules are negatively charged. So when they are passed through a voltage, longer DNAs will have a higher negative charge on them, and shorter DNAs will have lesser negative charge on them. So shorter, or we can say smaller DNA would come towards the positive voltage, and this process is called gel electrophoresis, which is used for filtering and sorting of the DNA based on their size. Okay, we just saw the Hamiltonian path problem, how it can be solved using DNA computing. When we consider this problem, since it contains only 5 points, so DNA computing does not looks very feasible for this problem. But consider a scenario where there are 100 points. Will it be feasible to solve that using the traditional semiconductor based systems? No, definitely not, because that amount of parallel computation cannot be achieved from semiconductor based computers, and it will never be feasible. In fact, there would be many problems which could not be solved by the present semiconductor problem. So for those type of problems which cannot be solved using the traditional semiconductor based systems, we can use DNA computing. Since DNA computing has a very high parallel computation capability. Now, let us discuss the disadvantages of DNA computing. DNA computing requires constant supply of proteins and enzymes, which are very expensive. Okay, and the second point is application specific. As I have already stated that we cannot use DNA computing in every types of problem, because simpler problems will be more feasible and more efficiently solved using semiconductor based systems. But for problems which cannot be solved using the semiconductor problems, for those problems DNA computing can be used. Small in size and power consuming. Since DNA computing is very small in size, so it is very difficult to manage and uh, use DNA computing. This can be a massive disadvantage for that, and it will require human assistance as well. The last point is simpler problems are more faster solved in electronic computers. I have already discussed this point. So, coming to the conclusion, DNA computers are still decades away from being able to compete with silicon based computers, but will eventually be more powerful than silicon based computers. DNA computers will be thousands of times smaller and more powerful. 1 pound of DNA has ability to store more data than every electronic devices ever made to date. DNA computers will be able to do many simultaneous calculations than silicon based computers because of its parallel computation capability. These are the two research papers from which I took a reference for this seminar report. Thank you.